Welcome to video 2 in B05, our Introduction to Coding with R. We talked in the previous video about the logistics of this course and learning resources, our aims, and the timetable for this. Now we'll talk about why you should be doing this and why you should have learned to code with R many, many years ago. So R is very, very useful. And we know for other areas such as physics, data management and ways of understanding data are far superior to biology, where things are underdeveloped. And then there's been a kind of a cultural limitation among biologists that we tend to steer away from courses in maths and statistics and computing because we don't think it's relevant for us. But that's not the case, especially now where we're going to engage more with larger and larger data sets and bigger projects. And that means we need to build the infrastructure for that, but also our own skills in genomics and bioinformatics. And there's huge opportunities there because many people haven't got proper training in that area. So let's engage with it more. R was developed first developed in 1993 by a gentleman in Ihaka. It's actually descendant of a language called S, which was developed in 1976. R is free and should be available on any device anywhere in the world, which is why we're learning it. It's something that you can use for years and decades to come, and is the standard language of bioinformatics and many aspects of computational science. S, if we look back at S, which is not free, it is originated between APL, PPL, and Scheme. That were the different ancestors of S. And then in turn, Scheme was actually developed from Lisp, one of the very earliest languages, and that in turn came from IPL, which was developed even earlier in the 1950s. So you can see this, the languages evolve, and R is definitely one that will be around for some time to come. So we know that large-scale data acquisition, molecular data, is easy, and that experiments are more complex. And therefore, our goal is to understand that we have multiple converging inputs, and we want better results from this myriad of molecular and high throughput data that's all over the place and very difficult to handle unless we really have the skills that are appropriate to understand it. And we have to think that we ought to use quantitative methods, the cutting-edge state-of-the-art quantitative methods, to understand our data and avoid traditional mistakes that are not appropriate. When we consider the scale of sequence data that's produced, and particularly the cost is the big thing that has changed over the years. So if we go back to 2001, the cost of producing megabytes of DNA fell rapidly until about 2007 or 2008, early 2008, and that scaled roughly with Moore's law. So Moore's law referred to the number of transistors that can fit on a computer chip, and that approximates compute speed in essence, and that has a doubling time of about 2 years. And roughly speaking, that holds more or less. And what we see with genome sequencing technologies is that there was a big revolution in terms of the application of high throughput sequencing by synthesis in 2007. The technology had been developed earlier in 2004, well, published in 2004, and indeed developed much earlier than that. But it became more commonly used there, and that's how you see this big fall in the cost, which has meant that high throughput sequencing of DNA, RNA, and other related nucleotides have become much more easy. And we know that if we go back further in time, that the amount of DNA sequence data available has increased dramatically, even without this genomics revolution. So that trend has continued throughout the history of bioinformatics and will continue further into the future. So we want to learn to code with R, and we want to think about how we can use it. So where does R fit? Well, number one, it fits with accessing data. So there's lots of data that's published, and you can write code to access the data without pointing and clicking at websites. You don't need to do that anymore. You can kiss that goodbye. That's not the way we want to approach things. You can be transparent. If your code is written down, you can show exactly what you did If version numbers change on a particular data source you want to access, you can change your version numbers and show transparently how did you do that. It's reproducible. You've written it with code, and if there's some sort of error or thing that needs to be modified, you can change that code and track what exactly happened along the way. So reproducibility stems directly from documenting exactly what you did rather than logging on to the likes of Genius or Prism or something, one of these horrendous tools that encourage people to make mistakes. Replication. You should be able to reuse your code for new data sets. So you might have initial data sets of high throughput sequencing, for example, and then you might find that the sequencing was okay, but you actually want to do it again because you have additional batch of samples coming in. You can reapply your code to that. So it's very, very versatile and flexible. So you might have this setup that you want to consider your bioinformatic experimental design, collect your data in line with appropriate approaches for your large-scale molecular data, or will let you clean your data properly. And we use those cleaning methods for other data sets that might be similar. You might use a tidy data structure, and we'll talk about that later as well. And then it'll allow you to make inferences about complex data sets. And that's really the important aspect of coding with R. What we want to get away from is these horrendous, messy spreadsheets of stuff linked together where people have color-coded their columns and you have it's utterly disorganized, and it's not machine readable, let alone even human readable. So we want to make sure that all our files are machine readable as well as human readable, and standardizing the file formats of the data that we use is very, very important. You see, there's all sorts of bad habits here, like weird symbols, spaces in the column names, all sorts of things that would just make everything very, very difficult. So we want to learn code with R because if you think about algorithmic complexity, there's ones that are difficult, like standard scripts we might write, or self-driving cars, or the Google search algorithm, which is very, very long. But then once you end up with these gigantic Excel spreadsheets, it can really make people's life very, very difficult. And we want to move from using that. These point-and-click tools are not the way to go. We want to embrace the future. So we need to move away from this idea that we can put things in Excel and write some functions that'll produce these awful dynamite plots, and that will tell us the answer. Or thinking that statistics is some sort of, decision tree where we can just choose which way to go. In reality, understanding your data is more complex from that. So we want to think about how we can use a variety of functions to understand our data and use the appropriate tools for it. So we now know that you may not need a script if you're doing something once, but in reality, you'll be doing things many, many times over and over again. So you need to write a script for that. So you can write your script, think about it, modify it many times. That will repeat many, many times as you generally make it better and better. Check it, test it, and then share it. So you will learn how to write your own scripts from what other people have done. And that's the culture: open science, fair practices, and particularly this idea that if you're trying to solve something, someone else would have solved it already 99% of the time. And if you look at packages that exist or what other people have done, you probably will be able to figure out what you need to do. It's a great example of this in the literature. There's many, many different examples. Here's an example of people that are looking at chronic obstructive pulmonary disease, and they're comparing an intervention group here versus those who got usual care. And we're looking at a survival plot of the time since discharge and the probability of having not having an event. So this is an example where manual records meant that these two labels got accidentally misused. because they were coded originally as 1s and 2s, and they got changed to 1s and 0s manually, which meant that the published paper, which had this plot showing that the intervention had had fewer, a reduced probability of no event, is more effective. However, in reality, this was not the case, that this intervention was unsuccessful, which meant that this paper had to be retracted from JAMA because it was fundamentally wrong. And this happens when you have analysis that are done manually and not with code. So what we want to do is to track how each result you have was produced, avoid manual data manipulation, version control of scripts and tools that you use. It doesn't have to be fancy, it just has to be tracked. You can write really, really simple code, and that's fine. That's exactly what I do most of the time. And as long as it works properly, that's the most important thing. Store your raw data separately so you don't touch it or manipulate it or do anything to it, and you have it in an organized fashion. And then also document what you do, comment as you go along. And if you look at the Galaxy, not so much the website or the computer service they offer, but the ethos behind that is a very, very good way to go. So as you might hear from me many times, we want to realize that certain approaches are maybe short-sighted, and that it can sometimes be problematic when we're trying to move on from old practices. But trust me, you can learn better ways of doing things. They will deliver you results. And with that in mind, you'll be able to pass on your new knowledge onto other people and develop a learning community around better practices. Learning to code with R can take some time. There's certain things that you have to consider for that. And sometimes you have to be curious, like a kitten, and change stuff to see what happens. Code is there to be changed, and you can cut and paste it and tweak things and just see what happens with it to understand it better. Sometimes you have to work hard and try stuff until it works. And that determination, I'm sure you have already. Googling the error message is often a good practice. You may not understand the error message, it may not mean much to you, but somebody else has probably seen it, and probably at this stage, millions of people have seen the same error message. How well you know the code that you have written is a philosophical question when you think about it more carefully, especially if you're using a variety of different functions. So don't feel you have to know every single thing about every part of coding or the code that you wrote. You just have to know how it works. So there's also the reality that you may use tools that other people have written on the basis that they know what they're doing. And being comfortable with understanding your own level of knowledge is an important feature of that. You will find that your initial attempts at writing or code are not great, but they're the first step before developing good code. So think of the first attempts at things as your way of understanding what path you should take for the final product. So what we're going to learn in this course are these things called ORMD files. We're going to learn about how to create an ORMD file and how to load them and use that as our major format for tracking what we're doing in terms of both coding and commenting. We'll look at how to install packages, particularly the Swirl package, and we use that tutorial for your own learning at your own pace whenever you want to. I will look at things like these awful dynamite plots we see in so many graphics, and we'll look at how we can do it better. We'll look at loading and viewing files as tables. We'll look at how to manipulate data using packages like dplyr and others. We'll look at saving outputs, and we'll look at using for loops as well.